Okay, so hopefully everybody is appropriately rested, woken up from the turkey and cranberry sauce, and you guys are, most of you are Southerners, so it's dressing here. I'm still a stuffing person. I don't know. We have a stuffing recipe that uses the King's Hawaiian rolls and portobello mushrooms and some other stuff, and I I won't have anything else. That's the best thing. So cornbread's good for cornbread. Anyway, um, so we're going to finish out chapter 17 today. There's not much left of it. And then I'm going to leave it there at that. So any time that's remaining at the end of the day today, we can use for review. Right? I'm going to then use the rest of today, like I have until next Tuesday to get all your stuff graded, and you have until next Tuesday to get all the stuff studied. It's good. What? I sent a remind and put a thing on. Okay, so the good news and the bad news is that the final is a week from today. The good news is you had so much more time than you thought you did. The bad news is that don't not use that time because you're studying for everything else, and then you get burned out and you have one day left to learn the genetic stuff, and we all know that doesn't work. So please keep working on this. I am available from pretty much every day from now until next Tuesday, so I can have multiple review sessions if you guys get with me. Generally, the genetics lab is open, and they finally got it so it's above refrigerator temperature in there. You guys found out the week before Thanksgiving. So, I can have multiple times there, or I'm happy to meet with you guys in my office too. If you can try to get more, if it's just one person, that's fine, but if you can try to get more than one person to come in at a time, that's better, because then your questions, even if the other person doesn't ask them, they'll, oh yeah, and we'll be reviewing stuff for them as well. All right, so how do we study for genetics? We talked about it at the beginning and all that, and then some of you guys believed me and some of you guys didn't. And then the didn'ts, stop. So whichever way you were, I want to buy something from you now, and it will be extra, everybody wants some extra points, right? So I will pay you 5 extra points. I'll put in like a bonus assignment up. It's not up yet, so don't go looking for it. And what I want you to do is more than 5 minutes worth of thought going into this. And what I want is for you to write like advice for yourself or advice for somebody on how to study for this class and do well that you've learned over the semester that you wish you would have known at the beginning. Like, what have you learned that works? Because I'll tell you that just like my kids don't listen to me and you don't listen to your parents, the students hear what I'm saying at the beginning and don't believe it, and oftentimes then there has to be some pain on the exams before things start to turn around. And so I'm hoping to use what you guys have written and sort of consolidate some of the things so that, and put it up at the beginning of next semester and use these going forward so that students can read what the students say you should do to study and so forth. So if you will do that for me, I will pay you 5 points. How about that? Is it a deal? Okay, so I'm going to make that do, because the final exam is on Tuesday, I'm going to make it do like Sunday night / Monday morning at midnight. So you have until the end of this coming weekend to get it done and turn it in. And please take some time on this because, like I said, you're not writing it for me. You're writing it for the intrepid genetics scholars that will be coming after you. And I'm hoping you guys can convince them, if they read what actual students have written versus me just blabbing about it at the beginning, that will be more convincing. All right. Oh, I already got that going. So, there's no lab today, so don't. You can show up to the lab, but I won't be there. Sometimes, I'm generally a nice person and I don't think I'm mean. You may have a different opinion, but sometimes I like to go down there and see who's there and giggle at them a little bit. But maybe maybe I'll do that, and maybe I won't this time, because somebody always shows up. All right, so we ended here. So we gave 4 different generic mechanisms of transcription regulation in prokaryotes in the previous chapter, and then by using the boy, my brain is fried on the cranberry sauce too. All right, let me try. I lost it. It's all right. We found out that they're not always, you can have, in addition to just a single repressor or a single activator, you can have systems where, like the lac operon, you have a negative inducible promoter system or operon system that also has an activator protein with it as well. And then now we're going to give an example of regulation in eukaryotes. Transcriptional regulation in eukaryotes is still a very active field of research. It's really, really complicated, and so I just want to make sure you guys all know that what we're going over here is a very generalized scheme to familiarize you with what's going on. So we've talked previously about RNA polymerase is brought in by TF2D with its TATA binding protein and the other little multicolored blobs here, the transcription activation factors. This sets up the polymerase at the transcription start site. And there's also this very large, not to scale here, mediator complex composed of at least 30 different proteins, which can be different in different tissue types or at different developmental times, that interacts with transcription factors that can be 75 to 100,000 base pairs away. And these can be either transcriptional activator proteins binding to enhancer DNA sequences. So the enhancers and silencers are the binding sites in the DNA. Enhancers bind transcriptional activators, silencers bind transcriptional repressors. Because DNA can bend and these sites can be so far away, including on the other end of the gene that's actually the part that's transcribed there, they act independently of orientation. So you can take this DNA out, flip it into the opposite orientation, and it will be fine because the DNA twists as a helix, many twists all the way up there, so it can interact just fine. We don't talk about that in this class, but here there are also proteins called architectural proteins whose job it is to bend the DNA like that, to make the enhancer or silencer sequence come into close proximity to the core promoter sequence here with the TATA box and where transcription is going to start. So our example that we're going to use for eukaryotic transcriptional regulation is GAL4. So in prokaryotes, E. coli was the main original model genetic organism, the one everybody was studying. So then you study it too, because there's a lot of work that's already been done and you don't have to reinvent all the tools. For eukaryotes, the model genetic organism is Saccharomyces cerevisiae. So this is the budding yeast that is used, different strains of it are used for raising dough for baking bread and other leavened goods, and also for brewing beer. This is different than the yeast that's used for wine, that's Saccharomyces ellipsoides. So just like E. coli, yeast, if they can have their preference, will like to eat only glucose, but they have the ability to eat other more complicated sugars. So galactose is a 6-carbon sugar, but its substituents are positioned differently than glucose, so you will need enzymes in order to convert galactose into glucose to use it. It would not make any sense to have those enzymes present if there's no galactose available to eat. So here's how it works. Here's the core polymerase machinery. The polymerase in sort of the whatever color this is, peach color there, all the transcriptional activation, general transcription factors. I put this down there because it's not shown in the figure in the book, but one of the galactose metabolism genes would be there to get turned on. So unlike prokaryotes, this is not an operon situation, so this same promoter setup will be used to drive multiple different genes, each of which is required for galactose metabolism. So here's how it works. There's the general transcription factors, the RNA polymerase shown in the peach, whatever that light tan color there. This is mediator. And then up here you have an enhancer sequence, and actually multiple ones of them. So the enhancer sequence in this case is called UAS. So UAS stands for upstream activation sequence. And all the upstream activation sequence is a specific DNA sequence that can bind the Gal4 protein. There are 2 of them there. This is a system in which sometimes there's 1, 2, 3, up to 6 different Gal4 binding sites, all within the same upstream activation sequence. And UASG just means it's for Gal4. Each of those will bind to a Gal4 protein. In this case, you got 2 of them there. So Gal4, unlike the allosteric proteins that were described previously, is not a shapeshifter. It does not change shapes. Gal4 is always able to bind the upstream activation sequence. And if it is by itself, it has, in addition to the DNA binding domain, it has a transcription activation domain that will interact with the mediator here and help with activating transcription. What turns this activation on or off is the presence of the Gal80 protein. So Gal80 is an inhibitor. In some cases in eukaryotic transcriptional regulation, the inhibitor or repressor protein will competitively bind to the sequence here. Gal4 or Gal80 is not binding to the Gal4 site and preventing Gal4 from binding. That's not how this one works. Gal80 binds to the activation domains of Gal4 to prevent those activation domains from interacting with and activating transcription down here. Okay? So Gal4 is always there, but it's blocked by Gal80, and that's going to be what gets changed when galactose is present. So we're going to bring in another protein called Gal3 that's shown here. Gal3 is a shape-shifting protein, as we'll see that Gal80 is as well. Gal4 is the only one that's not. Gal3 has a binding site for galactose, and it also has a second site that can bind to the Gal80 protein. But if there's no galactose present, Gal3 is in a conformation where it can't bind to Gal80. So Gal4, Gal80, Gal3 are sitting around all the time. The only change that's occurring is whether galactose is present. So if galactose is present, it will bind to Gal3 and change its shape. So now Gal3 can go and bind Gal80. So galactose binds to Gal3, that goes up here and binds Gal80. When Gal3 plus galactose binds to Gal80, Gal80 loses the ability to bind to Gal4, thus relieving the inhibition on Gal4's activation of transcription from genes driving galactose. So Gal3 you can think of as an allosteric protein that is an inhibitor of the Gal80 inhibitor, right? So I know in English class they told you never to use no double negatives, but here we're going to. So this one says no to Gal80, which was saying no to Gal4. So again, it's one of these, the enemy of your enemy is your friend situations. So Gal3 inhibits Gal80, which prevents it from doing its job, which is inhibiting Gal4. So you are relieving the repression of Gal4 and allowing the Gal4 activation domains to interact with the mediator protein and therefore turn on transcription. Make sense? So again, like I say for everything, don't go and try to memorize the list of proteins. Try to learn the story. Because if you can tell the story, then if you can explain how this works to somebody, then you need to automatically know how all the proteins work in order to do that. So you'll know how Gal4, Gal3, and Gal80 interact to get this going. Okay, so another feature of eukaryotic transcriptional regulation that is not present in prokaryotes is due to the fact that, like I said, these enhancer sequences can be very, very far away from the promoter. And that can be a problem because if this enhancer can interact with this promoter that's 100,000 base pairs away, why couldn't it interact with another promoter, that would be to the left if it wasn't bent over like this? If this was straightened out to the left, there could be another promoter way over here that's 100,000 base pairs on the other side, and Gal4 could interact with mediator for that one and activate it too. So how do we prevent this unwanted activation or repression from these factors that are binding very far away from the transcription start site? We do it with elements called insulators and insulator binding proteins. So you can think of this as either like, a wall around a castle or like a moat around a castle. If you put an insulator element in between two genes, anything on one side of the insulator over here can't affect anything over there, and anything over here on the right can't affect anything on the left. If you were to mutate this insulator, then you would have the potential for this enhancer 1 on the left-hand side to influence gene B, and this enhancer 2 on the right side to influence gene A. So sometimes before CRISPR technology, which allows us to just change the genome at will, you would make transgenic animals. So you'd be putting in a very large piece of DNA into usually mice, because that's the model organism that works for. Well, these introduced transgenic sequences would just integrate into the genome somewhere randomly. That's not a good situation, but it's the best we can do. And you'd have transcriptional elements necessary to drive transcription of the gene you're putting in, but you wouldn't want them to affect anything on either side of where this piece of DNA goes in the genome. So you would put insulator elements. If this is your piece of DNA between my hands, you would put an insulator over here and over here so that when this whole piece goes in, whatever you've put in won't affect what's in the genome on either side of where you put it in. That issue has been largely gotten rid of, like I said, by CRISPR technology, where generally now if you want to change a gene or something like that, you will just do it right where it is normally, the locus of the gene in the genome. We're not going to have time to talk about CRISPR in this class, but if anybody, like next semester or something, just come and talk to me and I'll be happy to go over it with you. It's really exciting technology. It's going to this is going to be the one thing that changes everything, because we can change whatever we want essentially in the genome to whatever we want at a certain spot. All right. So here's another example of regulation. So SV40 is simian virus 40. So I actually worked on this one a little bit as an undergraduate student. It's not used too much anymore, but it was used as a model because it's a very simple virus that infects monkeys. And so humans can get infected with it, but they thought at the time, and they've kind of changed their mind now, that it wouldn't have any negative effects. So this is SV40 is propagated by growing kidney cells that were isolated from a monkey in dishes in the lab. So no monkeys were, well, one monkey was harmed, but once you get the cells, now they're just growing in the lab. And this was used as a model for both transcription, DNA replication, and as is shown here, how you can get regulation of gene expression by alternative splicing. So the alternative splicing we've talked about so far involved, here's a gene with 5 exons, and in one splice you can use some of the exons, and the other splice you can use some other ones. What's different about what we've got here is these segments shown as A and B right here don't have an intron between them. So the coding sequence goes all the way from that end to that end. There is an intron between B and C here. So this gene is used to make 2 different what's called antigens. Large T antigen, which, surprise, surprise, is a bigger protein than small T antigen. These are used at different times during the viral life cycle. The way this happens is by picking alternative 5' splice sites. So in both of these examples, right here at the end of the intron, the beginning of exon C there, is where the splice acceptor is going to be. And the choice is whether the 5' splice site is here at number 1 or there at number 2. If it's here at number 1, then this whole piece, the coding sequence in B there and the intron, are all taken out, and you go right from coding segment A exon A to exon C there. And there's the messenger RNA, and you get the large protein. However, if an alternative splice is made, then the 5' splice site here, number 1 is not used, and number 2 is made, so all that's taken out is the intron. So now you get exons A, B, and C together, and you're thinking, what the heck? That's a bigger messenger RNA, right? Why does it make a smaller protein? The answer is not shown in the figure in your book, but I drew it in for you. In the middle of B here, there is an in-frame stop codon. So here, you start translation at the 5' end, and it goes all the way to the end. Exons A and C are both translated into the protein. However, over here, you get exon A is translated, part of this exon B is translated, but then you get a stop codon here. That's it. Even though there's plenty good coding sequence in the second half of exon B and all of exon C, transcription stops right there. So ironically, even though you have a longer messenger RNA here, you end up with a smaller protein. And I hinted at a little bit and said we weren't going to discuss it too much, but this is one example where I'm actually going to say that is the decision on which of these splice sites to make is made by these SR proteins. So SR just stands for a serine and arginine rich protein. So it has a lot of those particular amino acids in it. SF2 is just the name of the protein. If SF2 is present, it will bind to and encourage the use of splice site number 2 as the 5' splice site. If it is not there, then splice site number 1 gets used. So when it comes time during the life cycle to make the small t antigen, at that time, from another locus in the simian virus 40 genome, you would get this SF2 serine and arginine rich protein expressed. It will bind right here, and it will encourage the splicing machinery to use that as the 5' splice site and take out just the green intron there and not exon B in the intron, and therefore you will switch from making the large T antigen to making the small T antigen. Yes? How would I ask a question about this? Well, I could show this and say, you would have to know that what happens when SF2 binds versus not binding. I mean, you would just have to know that if you get, the alternatives are that you get this whole thing spliced out, then you get a protein that is composed of exons A and C, and that all gets translated. However, if exon B, or the second half of this exon, is left in because you pick splice site number 2 for the 5' splice site, then you get a larger messenger RNA but a smaller protein because you haven't included stop codon here. And the decision to do that is mediated by the presence of SF2. Yeah. I mean, just understanding about how it works. All right. So prior to CRISPR technology coming in, RNA interference was the previous next big thing. I want to point out that both CRISPR technology and RNA interference were discovered not by looking for new tools for molecular biology, but by trying to figure out how something in biology works. CRISPR is a defense mechanism used by certain bacteria. RNA interference is a defense mechanism used exclusively by eukaryotes. In this case, it is a defense mechanism to counteract viruses that may come into the cell. Some of the viruses have either single-stranded or double-stranded RNA. And a lot of the single-stranded RNA viruses can get converted to double-stranded DNA as well. Double-stranded long DNA pieces are not common in eukaryotic cells. Yes, RNAs can fold up to make complicated three-dimensional structures like we saw in transfer RNA, but the double-stranded parts are those hairpin sequences and help the RNA to fold into a three-dimensional structure. Here we're talking about a longer piece of double-stranded RNA that looks like double-stranded DNA that doesn't, it's double-stranded the whole way and doesn't fold up into a tertiary structure. So if you get an RNA, if you get a double-stranded RNA like this going into a cell, there is a protein in the cell handily named dicer for your learning ease. So what dicer does is it takes the long double-stranded RNA and chops it up into about 18 to 21 base pieces that are still, even though they look like they've separated here, that are still double-stranded pieces. So dicer does what the name suggests. It dices up the long double-stranded RNAs into smaller pieces. Then these smaller pieces, which are called siRNAs for silencing RNAs, will become part of this RNA-induced silencing complex, or RISC. So RISC contains multiple proteins and includes one of these pieces. So it will be one of the strands, not both of them. Some will be the strand that binds to a messenger RNA that was coded for by this double-stranded RNA in the genome. When you get that perfect match between the little piece of the siRNA that's bound in the RISC complex, it will base pair with the RNA, and the spot on the RNA with which it will base pair depends on where you're at. So it's this piece right here that's in there, so it binds. If it was this piece, RISC would bind right there. If it was any of these pieces up here that got into RISC, you wouldn't get any binding on this particular messenger RNA because these sequences up here match perfectly the sequence of the RNA there. Down here, you will get the reverse complement of the pieces. So if this messenger RNA is 5' to 3' left to right, this little piece of siRNA that's part of the RNA-induced silencing complex, this little guy down here is 5' to 3' right to left. And when I say a perfect match, it means all the bases here in this piece base pair perfectly with that. And that's going to be very important later. So what happens then is that when the RISC finds that, it chops up this messenger RNA, and then the messenger RNA gets degraded. Why? Because we're in eukaryotes now, and you end up with a 3' end here with no poly A tail, 5' end there with no cap on it, and the cell can recognize it as being defective and get rid of it. So this is the original mechanism that evolved for defense against viruses. Yes? With evolutionary mechanisms, you can't fabricate a whole system. You can only change what's there. And so what you're doing here is cutting up messenger RNA. So what happens with a lot of these double-stranded viruses is they are retroviruses, right? So this double-stranded RNA will be present, and then it will be made into DNA and stuck in the genome somewhere. Once it's DNA, then how this is going to just go cut up the genome? It can't. It would wreck it. It recognizes the double-stranded RNA when the viral infection first happens and gets this going, gets this response going. Then, once the double-stranded RNA gets converted to DNA and put in the genome, so first it can get this and chop it up. But second, if it gets into the genome, then sometimes even with so some viruses don't go into the genome too. This is going to work both ways. Sometimes the viruses don't go into the genome anyway, but you will get this converted to DNA without insertion in the genome, and in both cases, then you'll get a bunch of messenger RNAs transcribed from it. Killing this doesn't get rid of the messenger RNA, which is what's really getting translated into protein. So you recognize double-stranded RNA, which shouldn't be there, get this mechanism going, and then any single-stranded messenger RNA will get killed. Right, and everything. So this is highly simplified, you know, so that we can explain it. We can explain it to you and not. I mean, this could be probably like 2 weeks. This could be like 4 lectures at least of stuff that's been known. This was discovered in 1998, and then in the next 15 years, there's been a ton of papers on figuring out how it worked. And this, so what you really want to do is, here, this is the essence of it. What you really want to do is kill the messenger RNA. But how do you kill the messenger RNA that's from the virus that's invading the cell without killing all the messenger RNAs that the cell needs to live? The difference between messenger RNAs that cells of an organism make and messenger RNAs and RNAs that come in as viruses is that in this, the cell never makes this double-stranded RNA. So that's the trigger right there. This is, the double-stranded RNA is recognized as foreign, then it's cut into these little pieces which are used to go kill the messenger RNA before the proteins can be made. Good? All right. So that's a defense against outside invaders. This was then co-opted by some organisms, including everybody's favorite, C. elegans, right? For another use, for a regulatory use. So the C. elegans genome was fully sequenced in, I think, 1997 or something, somewhere along there. So the Human Genome Sequencing Project used C. elegans as a warm-up organism to learn and make mistakes because C. elegans has a smaller genome. So the human genome wasn't done until maybe 10 years later, like in the early to mid 2000s, so 5 to 10 years after that. But when the C. elegans genome was discovered, or was published, you could use computer programs to go through the genome and find transcription start sites that have, you know, that look like, hey, transcription could start here because there's a TATA box and other sequences that transcription should go, right? It looks like a legitimate promoter. Many of these drove genes that look like they could be made into messenger RNAs. Of course, then you get ribosomal RNAs and transfer RNAs, but everybody knew what they looked like. You would find a bunch of small genes in the genome that coded for RNAs that were definitely not messenger RNAs, definitely not transfer RNAs, definitely not ribosomal RNAs, but there's a ton of them. There's like hundreds of these in the C. elegans genome, and what the heck are they? The interesting thing, and again using bioinformatics and computer database searches of the genome, was they found out that every one of these little guys matched a sequence in a bigger gene that did code for proteins. Usually, should be down this way a little bit more, usually these little RNAs would be the reverse complement, so they could base pair with the 3' untranslated region of bigger genes that did code for messenger RNAs that would be translated into proteins. What the heck? They also found that these little RNAs, so if you stretch this out from left to right, it would go all the way over here. There was complementary sequence at the 5' and 3' ends of these little genes that not only did the little gene have a match to a bigger protein coding gene, but the little gene could base pair with itself to make this hairpin situation where you have a long double-stranded region and this little loop of the hairpin that was just to turn it around and get it going the other way. It turns out that these are being used by the RNA interference pathway as well. And once RNA interference was brought on board to combat viral infection, it turned out that the cell has got another use for it in gene regulation. Now, like I said over here, this sequence perfectly complements the messenger RNA of the virus that you want to kill. The reason is because it's cut directly from it. This guy over here is a microRNA, so siRNA, silencing RNA, that is cut from double-stranded by Dicer. Here you have microRNAs. These are in the genome, like I said, and make this piece that's about the same size as an siRNA. Dicer will then cut the loop off, and it actually cuts this little piece off of there too to make, and this is not correct, it will just make, this is going to be like one piece. It cuts here and it cuts here. Sometimes the artists get a little carried away and nobody checks it. There should only be like one piece that comes from this. This little piece gets cut off, that little piece but there's only one piece in the middle that is the microRNA. That will also go and be part of RISC, and it will go bind to the 3' untranslated region of some other gene in the genome. But there is usually about one base pair that's different, or one base that's different here. So you get an almost perfect match where it base pairs, but then right here there's only one base that's different, so it sticks out a little bit. And that's super important because, as we saw in the previous slide, if you have a perfect match with RISC, you get a cut and the messenger RNA is destroyed. Here, you don't want to do that. The RISC actually binds to the 3' untranslated region of the genes that are being regulated and sticks around there. So this inhibits translation because you can't get circularization of the messenger RNA. So this allows a cell to accumulate a lot of messenger RNA that it's going to need to make a whole bunch of protein, but not have it translated until right before it's needed. Right before this messenger RNA is supposed to be translated, another mechanism comes in and relieves that binding and gets rid of it. Now, with RISC and the microRNA not bound to the messenger RNA, translation can start. So this all began as a mechanism for defending against viruses in which you want to chop up and kill, right? And it evolved into, well, this is still there. Now you've got this other mechanism that involves microRNAs expressed by the genome for regulating your own genes. So you can have genes transcribed in places you don't want them to be translated and just sort of hanging around or get stored up, and then this repression is relieved later on. In some cases, and again, this is a big generalization. In some cases, that's all it is. In other cases, you get this kind of stopping the messenger RNA from being translated, and then this complex will interact with a methylating agent that will go back to the DNA that the RNA is transcribed from and methylate it. Remember, we talked about methylation of C's and CpG dinucleotides. That is a mark for inhibition of transcription. So here, in some cases, you just get messenger RNA stop, wait, don't translate it. In other cases, you are trying to get expression of a protein stopped immediately. So you inhibit translation and you go back then, and the same mechanism will cause the DNA for that gene to be methylated and stop transcription of it. This is highly simplified. So there are molecular mechanisms that decide whether it just does this and hangs around, or whether it does this to turn it off and does the methylation as well. Some organisms, I think, that don't have some organisms exist that don't have, I think we are one of them, that we do not do RNA interference, but we do have microRNAs. So the original use has gone out of us, but the alternative use is here. I think we have them. I'm going to have to look and make sure. All right. So once one of these phenomena is known about, then scientists immediately jump on it and try to use it as a tool. So once we figured out that RNA interference can work to cut up messenger RNA, we're like, well, if we want to stop expression of a certain protein in C. elegans or in tissue culture cells or whatever, we could do that by, we're not going to infect it with a virus, but we could make double-stranded RNA that looks like the gene we want to get rid of and make it be a perfect match so that you get the cutting mechanism and not the inhibition of translation mechanism. So what we've done here is we've taken a plasmid, scientists have taken a plasmid, so this would be a whole circle down here, it's just not shown. Grow it up in E. coli, which you guys have done, transform the plasmid into E. coli. We talked about transcriptional regulation in E. coli already, right? So because promoters are known, which promoters turn on expression, they've taken and put a promoter pointed in opposite orientations on either side of where you would put DNA that matches the RNA you want to get rid of. This is a man-made example of exactly what shouldn't happen. Remember I said you never get a gene on the top strand and on the bottom strand trying to be transcribed at the same time, except in some viruses, and then they're not at the same time, it's later. Well, here, because you have a ton of these plasmids and because they're in E. coli cells, even though the polymerases are going to be bumping into each other coming from opposite directions, you have this promoter over here on the left drives transcription using the bottom as template and making this piece that matches the top here, and you have this promoter driving transcription from right to left using the top as a template and making this piece that matches the bottom strand there. And because you have a ton of these plasmids, like a few hundred, in your E. coli cell, you get a whole bunch of these strands and a whole bunch of these strands that can come together because they match and base pair with each other. And so now you've got double-stranded RNA. And you can either take that double-stranded RNA and inject it into your organism, or in a really cool maneuver that the C. elegans community developed. Remember, I know, I keep bringing this up, everybody's favorite organism, right? The C. elegans, what did they eat for food on the plates? Those little spots were E. coli bacteria, weren't they? So instead of regular E. coli, you put little spots on the plate of these super E. coli that are making the double-stranded RNA. And the C. elegans will eat the E. coli, pop them open, and then the C. elegans gut will be full of double-stranded RNA. And it was discovered by accident that C. elegans have the ability to take up RNA through the lining of the intestine into the organism. And that will start an RNA interference reaction. So you can take a C. elegans worm that is wild type in the genome, and you can feed it bacteria that are making double-stranded RNA that match, say, the UNC13 gene that we used on chromosome 1. Those worms will have babies and they will all be paralyzed, just like they were UNC13 mutants. What? Like what? So even though they are plus over plus for their in the genome, like on the chromosome, all the normal UNC13 RNA that's being made, or enough of it, will be broken up by the RNA interference pathway that it will be like they aren't making any and they will appear UNC. And there is a mechanism in C. elegans that I don't have time to get into that keeps this going. So even if you stop feeding, so if you feed mama worm, she will have babies that will be UNC even if you put the babies on normal food. And their babies will be, I think you go 2 generations, and their babies will still be paralyzed. It won't be until like the 3rd or 4th generation where that RNAi pathway that got set up, it's almost like an immune response, will wear off, and then the worms will go back to being wild type. So this was before CRISPR, and so this was incredible because you could eliminate expression of any gene without messing around with the genome. And so I have in a freezer upstairs a library. It's a bunch of, you have the, you picked worms to 24 well plates, right? So I have 384 well plates, exactly the same size as the 24 well plates, but with 384 holes instead of 24. So just a ton of little, and each one of those has E. coli growing a plasmid with a different C. elegans gene. So if I want to knock out, pick a gene, right? I just go on my computer and look up in which plate and what hole it's in, pick the DNA there, or pick the E. coli, grow it up, and then I can feed it to the worm and knock out the gene that I'm looking for. So pretty incredible, except doesn't work in the germline. So all the sterile genes that I'm looking at, this doesn't work because there's another mechanism. There's another mechanism in the germline. All of this now is kind of a moot point. So RNA interference was really cool, and then it was a great tool for 10 years or 15, 10 to 15 years until CRISPR came around, and now we're like, forget that, because we can, if we want to make a mutation and knock out UNC13, we just go right into the C. elegans genome and knock the sucker out. It's absolute, the CRISPR stuff is just amazing. It's like, let's say Mr. Wizard stuff, but even I'm too young to know who Mr. Wizard is. So it's actually really cool stuff. But this was, and in some cases, in organisms where CRISPR is not an established technique, if there still are any, you can do something like this. And of course I have to put these guys up. So these guys published, each of them independently published papers in 1998 describing the RNA, describing what RNA interference is and correctly understanding that it's the production of double-stranded RNA, and they didn't have all that mechanism that I showed you worked out, but they had enough that that they showed how it worked, and then this whole field popped up and everybody jumped on and started working out what all the little individual proteins did. And Andy Fire, who was at Carnegie Institution of Washington, which is actually at, oh man, my head's not working today. And Craig Mello, who's at University of Connecticut, UConn Medical School. Andy Fire moved to Stanford then, because once you win the Nobel Prize, you can pick where you want to. You can pick where you want to live and how much they pay you. Johns Hopkins. Carnegie Institution of Washington is a little research unit that's located next to Johns Hopkins in Baltimore, Maryland. So these guys actually got the Nobel Prize in physiology or medicine right when I was finishing my postdoc and getting ready to look for my first job. But that was another thing, like the David Alice histone code thing that I showed you, where there was no doubt that these guys were winning the Nobel Prize. It was just when, and they got the Nobel Prize very quickly after, relatively. They got it 8 years after their work was published, except for like Watson and Crick. That's pretty fast. So if you want to get a Nobel Prize, you got to make sure to get it, and you got to live for 8 or 10 more years after that at least. So keep that in mind. All right. Well, that clock back there is wrong. It is 10:26. So that is it. And I am now available for, we can talk about anything we've gone over up to and including this point. And so there were a few emails that I got, and so here's the deal. If you emailed me asking about earlier stuff that hasn't been graded yet, there's a couple people who turned something in separately because they had problems with the computer or whatever. If that's you and you just emailed me, hang tight, and then I'm going to put out an announcement within the next few days that says, I think everything is, graded, with the exception of the lab reports, which are going to take longer. I saved those for last. Anyway, then let me know. And also I'll post everything on the lab website. And so just when I do that, then look back over and make sure, like, if you've emailed me something, it's been a weird semester. So if you've emailed me something, when I say is everything graded, please look in your grade book and make sure everything is there, and then say, no, I sent you this on, like on whatever day. All right, so so final exam Tuesday, December 10th, 8 o'clock to 10 o'clock. It says on the schedule 8:30 to 10:30. It's the same length as every other exam, so I've put in the syllabus and I'm holding it to like 8 to 10. You should be able to finish in 2 hours, especially this one, because it's like the previous one where there's not a lot of calculations that take extra time. But I'm giving you the 2 hours anyway because I'm a strong believer, and I don't want anybody to think that they could have done better if they had more time and they were rushed. So 2 hours should be well over plenty of time to get your stuff done. And yeah. You do not need a Scantron for the final, because it'll be just like the second exam. I'll give yours back and you'll start at number 51 on the other side. All right, so Sorry, Danielle, we're going to take your table on the way out. Well, sort of. I'll be available now to answer questions. Everybody should have their lab notebook here today, don't you? If you do not have your lab notebook here today, you need to run and get it and bring it to my office during office hours, because after that, that's it. It's late. So on your way out, just sit them on a table next to Danielle, and sorry, it'll take up a little bit of space there. Oh, the other thing, the emails that you may have gotten about the class evaluation or whatever, if you have not already done it, the email they sent you is not correct. You have, I believe, until the end of the day tomorrow, the 4th. You have until final exam start. If you have not gone on and done a course evaluation for this class and your lab section, which are two separate things, please go and do so. And fill in the little bubbles with the numbers and all that. But please let me know what you think in writing as well, because that helps me more than the numbers. Things that you think could be done. Hey, if you did it this way, I think it would be better. Like if you're going to have suggestions that actually have something that I could do, those are good because I could work on it. I think you should have 50 extra points of extra credit is not, that's not it, but or especially if there's a certain way I do something that you like, or this worked, but I think you could do this other thing a little bit better. Those are the kind of suggestions that I actually read these and try to figure out how to improve things. And a lot of the way I do stuff now is due to past suggestions. So please take the time to go on and do that. I know you're really busy with finals, but even typing something out to answer the question shouldn't take all that much time. Okay, so get with your friends or whatever, and I can meet with you anytime you want, any of these days. So Wednesday is tomorrow. Wednesday is a good day, or Friday is a good day, because nobody will have exams on those days. I can also meet on Monday, but some of you might have exams during that time. And as with the previous review sessions, I will record them if you absolutely can't come. But if you can be here at all when we decide when the review session is going to be, please do so. It's much better if you're here in person and can actually ask questions and interact with me than just watching the video of what everybody else has said. So any questions now, or should we just let it go and then come back? All right, well, I will see you at the review session, hopefully. And then for the final exam, please don't forget to put your lab notebooks back here at the back.